at University College London. His current studies are at the forefront of a rapidly evolving field of investigation into the role that immune response plays in neurodevelopmental conditions such as autism. He was the first to demonstrate links between immune dysfunction and the severity of impairments that are hallmark features of autism, such as social interactions and communication. Overall, Dr. Ashwood has worked across traditional disciplinary boundaries to examine connections between different biological systems during development in order to understand how they lead to the characteristic features of autism. Specifically, he has highlighted the importance of innate immune pathways, gut immune brain connections, and the presence of autoimmunity in some children with autism. He's the author of over 100 articles on autism and has received recognition for his innovative work. Dr. Ashwood, please take it away.
Thank you very much, Pramila. Let me just share the screen. Okay, thank you. And, where am I? There we go. Oh, jumped ahead. All right. Well, thank you very much. Thank you, Pramila, for your introduction. I'll make sure I cut back the bio next time because that was too much. Thank you. It's an honor to be able to speak at the Brain Foundation. And give a little update on some of the information that we're doing. So today I'm going to talk about immune regulation and its issues in gastrointestinal symptoms in children with autism. Okay, there we go. And so we've heard from the excellent speakers this morning about gene and gene environment interactions, and I think from Sarkis and Joe about and Matt and Stuart about the fact that there's a very strong genetic link which links with high risk genes, and then a combination of maybe lower risk genes that all contribute to potential risk for autism spectrum disorders. And the same can be true for the environment, that there's potentially very high risk environmental agents that we, at this point, we don't really know. Maybe valproic acid is one that springs to mind, but a number of others that may be, common environmental exposures, such as air pollution, changes in the microbiome, or like with the genes, you can get combinations of the two. So you can get infection, and you can get maternal infection, for example, in some of the animal models that Sarkis mentioned earlier, and the microbiome. So you can get combinations of the environment and combinations of the genetics, and somewhere in between, maybe this is what is the most likely, or at least in my opinion, the most likely causes of autism spectrum disorders. And at that interface, you have maybe the immune system. It's uniquely situated where it deals a lot with environmental exposures, and is, of course, most of its function is predicted by genes, and changes in gene polymorphisms or epigenetic dampening down of gene signals can change the immune function. So this is where I've always been interested, but I always want to make sure that we start this any talk with the fact that the immune system is incredibly important, and it's important to mediate resistance to infection or to eliminate infection. In fact, I guess we all know that now because we're living in the age of COVID, and the importance of protection against COVID, and the development of things like herd immunity, so that the more vulnerable in society are sort of protected against that. But we have this complicated and complex system where a lot of cells from the immune response interact to eliminate and protect us from things that are potentially harmful and potentially fatal. So it's always important to think about it in that way. But when things go wrong, you can get immune responses. And again, that become apparent, you become problematic. So again, if you think about COVID, this idea that there could be a cytokine storm, the release of mediators that cause further damage or the most damage to an individual, and could lead to progression of disease, of the infection. You also to the fact that some of these immune mediators could lead to issues such as the long-term COVID, where you get fatigue and all sorts of extra chronic symptoms that can last for a very long time, and we just don't know how long that is. But when thinking of autism, we can see when things go wrong, people have shown evidence that there is inflammation in the brain. Andy Zimmerman and Carlos Pardo produced some of the first work on that, very important work on that, about showing that there were increase in cytokines in the brain in individuals that had autism. More work, recent work has shown that there's cells of the immune system in the brain, the microglia, that they have become activated and that they can produce factors that could attribute to changes in neurological function. We have a speaker, Dr. Fasano, later has also shown that there's altered blood-brain barrier function in the brain, and we had a speaker last year who's talking about the fact that they could be T cells that come from the periphery that get into the brain, and that that could then lead to a progression of sort of inflammatory responses. We also know there's a lot of common comorbidities, immune comorbidities in autism, such as allergies and asthma, and in particular interest to me is things like GI dysfunction. And again, I don't know if Dr. Kriesman will talk about it more, but the increase of inflammatory bowel disease has now been set as the population of children with autism age. The prevalence of inflammatory bowel disease in the autism population has been shown to be increased. When thinking of immune cells and immune function, we've seen that there's differences in antibody levels, there's differences in monocyte cells, some of these other immune cells that are very important for dealing with different functions of infections or involved in asthma and allergy and other things. They've all been shown to be dysregulated. And I think many of you probably know that there's this increase in antibodies directed towards proteins of self, so your own protein structures. So what I'm interested in is how potentially the balance in the immune system, if it shifts one way or another, how does this affect the nervous system? And we've shown that sometimes increased inflammatory cytokines, this has been linked with worse behavior. So there's increased, so as these inflammatory cytokines increase, social behavior gets worse, communication gets worse, and we often see more increased restricted repetitive behavior. So, but that's not always to say that increases is the only way. You can also get the fact that the immune system can decrease, and this could also have effects on the nervous system. So some immunodeficiencies could also have a serious effect. So you just got to get it right at that tipping point, and otherwise the two systems can interact and change. And also the nervous system can have a big impact on the immune system through production of neurotransmitters and and signals that can tell the immune system whether to activate or not. And this is maybe how things in the microbiome can be producing neurotransmitters that affect both the nervous system and the immune system as well at the same time. So as an immunologist, why is it important for me to sort of think about mucosal immunology in particular? And this is how I first got into the field. And it's the most immune cells in our body are within the GI tract. So this is something that is you have more immune systems in follicles, you have more immune cells within the lamina propria, and this outweighs cells in the rest of the body. And what to me is very unique is that there's a separation of what's going on in here in the lumen of the gut, which is bits of dietary factors, bacteria, and all of these immune cells that are present underneath that one single cell layer of cells. So it's not a big barrier. It's just there are some protective reasons why this barrier is effective, but it's not very. The difference between host and environment is very small. So this to me has always been a fascinating thing to look at, and also to me is why don't we react to everything that we take into our body, because it is essentially a foreign element that comes into our body, and there are checks and balances in place to protect us from reacting to all of these things. So that's one of the ways that I look at these things, and how I've always been interested in it. And then when I moved from looking at Crohn's disease into looking at GI issues in autism, at that time there were a number of early studies, and the first study is really to go back and look at the original description of autism. And if you look at that, you'll see that at least 8 of the original number of subjects that were described, at least 8 of them had issues that could be described as GI issues, such as vomiting, eating issues, food issues, selectivity, and so it was always there. And then further over the years in the 70s, people started looking at things like glycosylation and showing that there's differences in sulfation. So some of these things could be related to the microbiome, or they could be related to the host themselves. Bacterial overgrowth, diversity or lack of diversity of microbes are certainly things that we've seen more and more frequently. We haven't really got a consensus of which bacteria are different, or which groups of bacteria are different, and that could depend on dietary factors within different populations as they're measured, or could also relate to picky eating in certain subjects, and selective eating or food sensitivities in certain groups. But we've also seen that positive interventions, dietary interventions, could lead to positive outcomes in certain patients. So the estimates of GI issues vary quite dramatically from whether you're looking at a population-based study or whether you're looking at a clinic-based study. But it's very clear that a good proportion of children with autism have some sort of GI issue. And I think when I first started this, 20 years ago, people didn't believe that, and now I think it's pretty well established that a good proportion, a third to a half of people, depending on which report you look at, have GI issues. And the main complaints are things like abdominal pain and constipation and diarrhea. And I'm sure Dr. Kriegsman's going to talk a lot more about this, and especially these sort of findings here, when this is the ileum. And this is a look from a colonoscopy, that they've seen that you get these formations of what's called lymph node hyperplasia. So these are lymph tissues that have expanded, and within that, a lot of T cells and B cells that are expanding and overreacting and over-responding and proliferating. Also, this has been seen in the colon. You see these quite distinct, these sort of responses. And then if you're looking at H&E staining, you'll see there's a lot of activity going on there with a lot of immune cells. So some of the really early work that we did, God, that's nearly 20 years ago, that we looked at, it was taking sections of the gut from individuals that had autism and typical developing controls, and seeing how many immune cells were in that tissue. So if you're looking at a typical developing child, you'll see that there's some. These are protective, and they're there to protect the host from what's going on in the lumen. But in the child with autism, there's a lot more of these immune cells stained in brown here. And that there's, when you look at the levels of these cells, that they're increased above the controls and similar to established inflammatory disease. They're not the same, but they definitely an increase in these cells. So when we looked at individual, these markers just denote T cells that are present, and we found that in different compartments of the gut, lamina propria, which is that level below the epithelial layer, and in the epithelial layer, that there's an increase in the number of immune cells within the gut in autism over typically developing controls, and same as inflammatory celiac disease. And this we found in a number of, we found it all the way through the gut. So we found it in the esophagus, we found it in the stomach, we found it in the transverse colon, and then the intestine. So it seems that it's not distinct in certain regions, which other things like celiac disease and inflammatory bowel disease are sort of contained within different regions within the gut. But this was a pan-enteric sort of inflammation which was subtle, but it was definitely an increase in those immune cells. So the next phase of, well, we've got all of these immune cells and we've got an increase in these immune cells, but are they actually doing anything? Are they producing anything that could be harmful or could lead to this inflammation that potentially we're seeing? And here, again, in some of the early work, we looked at whether they were producing different types of inflammatory cells. And, you know, the immunologists like to divide things and like to group things into different categories that not always fit easily, but you know, to help you differentiate what might be a certain process in the immune response. So here we looked at things that were Th1 cells, and this is thought to be involved in inflammation and cell-mediated responses, and also for Th2 and regulatory T cells that are involved in potentially, if the increase in Th2 cells, they're involved in sort of allergy, asthma type responses. So what we found is that in children with autism, so in the yellow, we had an increase in these in pro-inflammatory cytokines. And again, because of things like COVID, we got more familiar with some of these terms, but like IL-6, this is an inflammatory cytokine, and TNF-alpha, another inflammatory cytokine. And these were increased, and they were increased not to the levels of Crohn's disease, and that's very important, but they were increased above non-inflamed controls. So this is actually terminal ilein. So the children who are coming in, who are the controlled children, they're coming in with complaints of GI issues and potentially they have inflammation. But these were controls with that had things like constipation and functional GI disease, which seemed to be an appropriate control at the time. But we also saw an increase in these Th1 cytokines, IL-12 and interferon gamma. What was interesting is that when we looked at the cytokines that are involved in allergy and asthma, we saw that there was an increase in these, which wasn't there in the Crohn's disease, such as IL-4 and IL-5. But when it comes to the regulatory cytokines, such as IL-10, we got this massive decrease compared to both inflammatory bowel disease and control. So what I took away from that and what we thought was that there's an increase in inflammatory, but a decrease in this regulatory signals. So what we also found is that what this may mean is that we've got this increase in inflammation, that it could have an effect on the barrier function. So we looked at the barrier, and then one of the ways we could look at is whether there was an autoimmune reaction at that barrier. So we have antibodies, IgG, that bind to we found that bound to the inflammatory, they're bound to the basement membrane of the epithelium. C1q, you just heard a little bit about complement proteins. When these two combine, that they form an attack complex that can lead to tissue damage. And what we found is that when we co-localize this on a biopsy from a child with autism, that those two coexist and they co-localize on that barrier, potentially meaning that this could affect the epithelial barrier function. So we're very interested in how this this could develop, and you know, this was work that I did as a postdoc, and I wanted to carry it on. And here we're looking at, and the hypothesis was to do with this idea that there is increased inflammation, but there's decreased regulation. So we wanted to look at the tipping point between these two things of what, whether in children with GI issues, whether there was this increase of these certain cytokines and a decrease in the regulatory certain cytokines. So it's that regulation that I'm really keen on. So we looked at different groups, autism with GI issues, autism without GI issues, typically developing children with without GI issues, and typically children with GI issues. And this is work we did with Sarkis and Alicia, who's going to talk later, and Paul Patterson. And so we started looking at what we'd take the blood, would isolate the plasma from the blood, would isolate immune cells, and we can stimulate those cells to see what they do. I mean, it's interesting to see a cell at rest, but it's also interesting to challenge it and see if it has a different type of response. So we could stimulate cells, see what cytokines they're producing, and also start looking at flow cytometry. And flow cytometry allows you to look at, does that individual cell change? Does it have certain things on the surface that shows it's activated or quiescent? So what we found, which was interesting, is that when we started looking at groups, when it came to pro-inflammatory cytokines, that we did see not only differences between children that were typically developing with no GI issues, but we started seeing potential differences between kids without GI issues and kids that had GI issues, that there were differences in pro-inflammatory profiles, but generally autism was more different than the typically developing children and had more activity to their once they were stimulated and then produced. Potentially more interestingly, when we looked in terms of the GI issue, when we looked at regulation, we found that there was a big decrease in the amount of this cytokine that's very important in immune regulation, transforming growth factor beta, that it was decreased in the ASD GI group compared to the ASD no GI and the typical controls. And that's at rest, but when you also stimulate them with different activators, so activates T cells, this LPS is important in responses to the microbiome and bacteria, and you found that there was this big decrease in these ASD in the ASD group. So we also wanted to keep going forward and looking at different types of cells, like we mentioned earlier. I brought in the idea of different Th1, Th2, and regulatory T cells. And what we found is that there was an increase in the group with ASD GI there was increase in these Th17 cells. Now, Sarkis alluded to the maternal immune activation model and how in that immune cells are important, maternal immune cells are important, and also cells in the offspring that could lead to GI issues. And what's interesting is that the paper, the most recent immunity paper, has shown that there's this increase in this animal model as well, which is very interesting, of Th17 cells. And we found the same here in humans, that there's this increase in Th17 cells, and also Th17.1 cells, which are those that produce IL-17 and also produce interferon gamma. So we're getting some parallels between an animal model and humans. So we also then looked at whether there's a decrease in these regulatory cell subsets by flow cytometry. And here we saw, again, that there's this big decrease in the ASD GI group compared to typical developing controls. What was also interesting is that we did see differences in behavior, and I'm sure that Arthur, Dr. Kriesman is going to talk about that as well, that in the ASD GI group, there's more, it seems that there's worse, more impaired behaviors. And we saw that these were linked to immune responses. So to me, what's really interesting is this key link between immune activation and, more importantly, immune regulation. And I think that's where things sit, that there is a decrease in this. It's important to maybe up the regulation to be, because at the moment it's way too low. So one of the projects that the Brain Foundation is supporting, and thank you for that, is that we're going to look at this epithelial barrier. And as I say, to me, it's amazing because it's just this one cell thick barrier, but it's very important how it, and there's various ways of how it can educate this regulatory immune cell population. So we want to look at this epithelial cells and we can take them from individual patients and we can grow them out in epithelial organoids, much like we heard discussed about cerebral organoids. We can take the immune tissue out, and we can look at the inflammatory responses. We can look at cellular stress responses, and again, look at what would happen when we challenge them with certain things like bacteria, what happens when we challenge them with different metabolites, what happens when we challenge them with different microbes. And so here we can have this, we can take the immune cells and grow them. And it always fascinates me that you can get these amazing pictures that look a little bit like the villi that you've been produced. And we're focusing on the ileum, terminal ileum, because that's where we think a lot of the immune responses are occurring. So we've taken a lot with the support of Brain Foundation. We've started biobanking certain pediatric patients from autism and from controls, and we've also taken some different cell lines from immune activation models and also some other models that might be important in autism, and the CHD8 model is important with GI issues and with immune issues in autism. We're going to run some single cell RNA-seq because there's going to be different types of immune cells within those populations of the organoids. And like I say, we're going to see what happens when we co-culture with different, from the same individual patients, co-culture with immune cells, with immune products, also with bacteria, if we can find a way of growing up bacteria without losing too many of the species within, when we sort of grow them out from patients, and see what the responses are of those epithelial cells. So I think this is very exciting, and hopefully that's where we're going to. This is the direction we're moving in, and I'd like to thank people in my lab. I'd also like to thank my collaborators, and I apologise for not putting on your names, but for Sarkis and Alicia, and we've got projects that hopefully we can get off the ground with Dr. Kriegsman and Dr. Walker. And of course, I'd like to thank the Brain Foundation for their support. So thank you very much. And can I stop sharing? Stop sharing. Okay, great.
There is one question on the chat. I think it asks, the anti-TNF drugs are used to treat Crohn's GI. Do they work in ASD?
I think that's probably a question for Dr. Kriegsman later, and it would be, you know, it's one of the things that I mean, one of the offshoots, I think, of the COVID crisis is that there are a lot of antibodies or antibodies out to different types of cytokines. So there's definitely a lot of now that there's these reagents that could look at how to change the immune response and immunosuppression based on specific cytokines. So yes, I mean, whether TNF is the right target is something that we have to look into. But I think, in my mind, I think we need to increase the immune regulation, and there's ways of doing that in terms of that you could potentially take sort of cord blood, for example, if that's stored, and grow regulatory T cells from an individual to be able to then give back. Let's see what else. There's a question. Is there a test that you can quantify base inflammation in a child? So again, there's not necessarily a, people have looked at that. I mean, what we haven't been able to look at is whether there's levels of cytokines and whether there's a way of looking at population normals, if you know, normative ranges. And so things happen in ranges, things happen at different ages. So different age, you're going to get a different immune response as you go through. So we need to have, if we're going to do that, we need to have larger subject numbers to be able to have something that's informative enough. And I worry about, you know, it's the same with any of us having a blood test, is that it's a snapshot in that time, and that you need to probably look at a longitudinal effect of what your T cells were, what your cytokine levels were. So we're kind of a long way away from that. But yeah, that's an interesting question. Okay.
I think Dr. Kriegsman raised his hand.
Sorry, once you start looking at, there's the Q&A, and I haven't got to the chats yet. Arthur, did you want to say something?
Yeah. Okay. Can you hear me?
Yes.
Yeah. Okay. Great to see you, Paul. Okay. First of all, thanks for the lecture. But just a basic question that I have is that, why is there no commercially available test that clinicians can use in advance of using a biologic agent like the TNF-alpha antagonist? Why is there no biologic test that we can do to identify those patients that do have elevated TNF-alpha versus those that have a more of a predominant IL-12, IL-23 type of profile, since there are drugs that are currently available that would be more effective in one versus the other? Why is that, at least to my knowledge, still relegated to a laboratory investigation and not commercially available?
Well, I think it comes down to the fact that there's nothing in the blood necessarily that in plasma or sera that's an easy target to look at. I mean, most of what we found that gives you the best differentiation between controls and individuals with autism is that you need to do something with the cells. You need to stimulate the cells and then be able to look at the stimulated responses. And I think what's interesting is that even within that, you have, and I hinted on it, and I did, maybe next time I'll show some more pictures, that with kids with GI issues with autism, they have this profile where they have increased IL-17, whereas kids without GI issues and autism, they have an increased IL-4, IL-5 profile. So you have a Th17 response and a Th2 response, but they're both up compared to controls. So, we just, again, it's one of those things that I think we just need bigger numbers. And, it's the same with how anybody established any sort of baseline levels of just what's high CD4, what's high, T cells, because they were able to measure quite a large number of a group before. And once we've got that, then maybe, maybe you can move on to the next thing. Or, hopefully we can do things like we can create certain things in a dish from iPSCs, let's say, and then you have a mini gut that you can be able to say, here's a response, and that's something that then the clinician, it will inform the clinicians a bit more.
Interesting. One thing that Dr. Walker and I are working on, and I don't know if he's going to mention this today, is we do have an organoid biobank of enteric tissue. We've recently started that. You may be aware of that, actually. And the other interesting and clinically relevant issue is that I would imagine that if there were a repository of, say, 1,000 ileum specimens from 1,000 ASD patients with GI inflammation, that perhaps I'd imagine would be, and versus controls, that would be a great starting point to identify which pro-inflammatory cytokines would be best targeted, either by existing pharmaceuticals or ones yet to be invented.
Yeah, I think that's definitely something that we need to further work, and hopefully the things that we might work on together will help us get some answers to that.
Looking forward to it.
Okay, so
we can move on.
Pardon?
We can move on to the next.
Yeah. Okay. What I'll try and do is answer the questions offline, or I can type answers into certain things as replies. So I have no idea who the next speaker is. I've never seen them before in my life. Obviously, sorry, poor joke over Zoom. But yeah, I've known Dr. Kriegsmann for a number of years. He's one of those pioneers, if you like, that have, and he, there's been a lot of obstacles that he's overcome, but he has persevered, and he's fantastic and brilliant, and one of the few pediatric gastroenterologists who are willing to see children with autism and to investigate the GI inflammation. Not many people do, not many pediatric gastroenterologists do, but he definitely, Dr. Kriegsmann, has been a major pioneer in this area, and has, as I say, there's been a lot of obstacles and issues that he's had to come through, but he has not only done that, but he's presented to the US Congress about his findings, and presented at major meetings, and presented to influencers, and has hopefully, he will be now, some of the work that he's been doing over the few years will finally, people will take on board and come along with him on some of the things that he's been trying to pioneer. Dr. Walker, environmental sort of interactions in the intestinal tissue, and again, they've had problems with trying to get obstacles through the controversial nature of some of just work in autism, work in GI issues, and I'm always very appreciative of just how clear they've been in the fact that this is very important work to get on with and to do and actually go out and do it. So I both think that they're amazing. So anyway, thank you.
You can go ahead and screen share.
Okay, here we go. I had to find it. Okay, can you all hear me and see me?
Yes.
Okay, great. Paul, I want to thank you so much for that wonderful introduction, and just to reciprocate your work. I didn't know that you were a postdoc at the time of those early publications. I thought you were already an established expert. But when I was just beginning working with these children about 20 years ago, it was your work on the slides, some of which you've shown today, that really gave me the foundation and the basis to move forward with what otherwise would have been a very difficult work. So I want to thank you for that, and thank you for allowing me to pick your brain over the years. And now we'll get on with the clinically relevant information that has evolved from Paul and others' work on the gastrointestinal tract of children with ASD and chronic GI symptoms. So what we've done over the years, and other groups have done over the years, is to identify and characterize the nature of the inflammatory bowel disease. But to date, and one of the panelists asked the question, you know, do TNF-alpha antagonists, do they help treat ASD inflammation? And the answer is a resounding yes, and we're going to get to that. But until now, no one has demonstrated the effectiveness of any medication on ASD GI inflammation. I've been treating it for years with a recent support for the Brain Foundation. Dr. Walker and myself have began putting together data, treatment data, to show, well, we're going to show you about that in just a second, to bring it into more of a clinical realm. So for those of you who are a bit weary of the scientific slides, we're going to show you some more clinical slides, and then Dr. Walker will follow me with the stats and the data and back to the science. Is that a better view?
No, the other one was.
The other one was? Okay.
This is good.
Okay, very good. Okay, so what we've termed the inflammation of the gastrointestinal tract in children with ASD, chronic GI symptoms. It begins clinically. It begins with having a child with ASD, and the confirmatory tests are in the realm of behavioral pediatricians. We're not going to get through. We're not going to go through that now. But as far as having GI symptoms, patients that appear in my clinic have an established diagnosis of ASD and a constellation of symptoms, and they can include any or all of the following: diarrhea, constipation, abdominal pain, failure to thrive, weight loss, vomiting, food refusal, episodic food refusal, where they won't eat for a day or two, and then they will eat, and they won't eat for a day or two. Abnormal stools, in general, very malodorous, strange color, and food intolerances. And other lectures I've gone through that in slides what they look like. But when a parent sees this, or a caregiver sees any of these symptoms in a child with ASD, those are the symptoms that mandate a clinical evaluation. And as you see on the bottom, parents frequently report that the GI symptoms that these kids have, the constipation, the abdominal pain in particular, those impact the quality of life of the child and the family unit more than the autism and the behavioral abnormalities of autism. And that's a very striking thing for a parent to say.
Excuse me, did you want to turn on your video?
Okay, there we go. Thank you. Okay, so the diagnosis of an autism-associated enterocolitis, and in a moment we're going to go through why we call it autism-associated, what distinguishes the inflammation that we've been speaking about and that Paul spoke about, what distinguishes this GI inflammation in autism from Crohn's disease or from other GI inflammatory conditions. And we're going to get to that, because those were the topics of previous papers of myself and Dr. Walker. And how do you make the diagnosis? So when you have a patient with ASD and have chronic GI symptoms mentioned above, you can see that they grossly at colonoscopy, you'll see, these are both pictures of an abnormal colon. Normally it should all be pink. You should normally see blood vessels like you see there. Those should be numerous blood vessels. And here we see really it's a light red, not pink, and it's also absent of any vascular markings. Here you see it's less involved. There's more pink, there's patches of red, some blood vessels. On biopsy, you'll see areas of chronic inflammation. You'll see crypt branching, both of which are seen in Crohn's disease, neither of which are diagnostic or specific for any particular the inflammatory disease of the intestine, but it's commonly seen, and most commonly seen in Crohn's disease, and it's commonly seen, very commonly seen in the autistic children as well. When we go during the colonoscopy, if we advance the scope a bit further, as you saw in the previous lecture, you see terminal ileum, which is the last segment of the small bowel, and you see a very marked proliferation of lymphoid nodules, which in and of itself is not inflammation. It does contain B and T cells and other cellular subtypes that are inflammatory, but on biopsy, we know from a clinical standpoint that this is something which needs to be treated because you have what's called ileitis, when these white blood cells infiltrate the epithelium of the colonic crypt. So these are all findings that we often that we see to make the diagnosis, the gross endoscopic diagnosis, and also the histologic diagnosis. When we do an upper endoscopy, I can see the esophagus, you can see the stomach, and the first few inches of the small bowel. When you do a colonoscopy, you can see the entire colon, and you can also visualize the last few inches of the ileum. But there's between 9 and 10 feet to 13 to 14 feet of small bowel that's all coiled up, that's not accessible to the scopes. Capsule endoscopy, or known as the PillCam, which many of you are not familiar with, is able to visualize this small bowel. It takes 8 photographs per second for a total of 8 hours. And when we examine those videos, we will see aphthous ulcerations of the jejunum. Here we see the more typical ulcerations or erosions or inflammation is this one, where it's not quite an ulceration that has exudate coming out like you see here in these photos, but it's more of an area of erythema surrounded by an area that has some mild lymphoid hyperplasia. This is what we see in the small bowel at capsule endoscopy. Overall, without going into it, other speakers have, there is definitely strong clinical cellular molecular evidence that ASD enterocolitis is distinct from Crohn's disease, but there's also a significant clinical cellular and molecular overlap between the two disease entities. So you saw some of those slides in Dr. Ashwood's lecture about how on the cytokine level, the cytokines, both intraepithelial and also systemic, are raised above the level of normal non-ASD children, but don't quite reach the level of children with without ASD and with Crohn's disease. What we did a number of years ago to demonstrate this is to do a PCA of the plotting of the ileum and also of the colon. This slide is of the ileum, where we have the Crohn's patients in blue, the healthy non-ASD, non-GI symptomatic patients in green, the ulcerative colitis, non-autism patients in this olive color, and the ASD patients in purple. And as you can see, the ASD has a clear overlap from a transcriptome point of view. It has a clear overlap with Crohn's disease. There's also a subset of the ASD and Enterocolitis patients who are kind of off on their own, and then there's only minimal, statistically insignificant mixture with patients who were healthy. So this provides the molecular evidence from a gene transcript level of a different yet very similar transcript profile of the ASD and enteritis of the ileum. And here you see of the colon, it's almost an exact replica of what we saw on the ileum. We have the healthy patients without autism, without GI disease. We have the Crohn's patients in blue. We have the ASD patients in purple, many of which overlap with the Crohn's patients, some of which are off on their own. And again, so we're dealing with a disease that's similar yet somewhat different than Crohn's disease. The immunopathologic nature of ASD enterocolitis and ASD is well established, and Dr. Ashwood went through those and other speakers as well. And as an inflammatory disease of the bowel, it is therefore logical, and this goes back to the question that one of the panelists asked. It becomes logical at that point as a clinician. What do you do with these patients? I have a patient who doesn't have what appears to be Crohn's disease, but clearly has inflammation of the gastrointestinal tract, and more importantly, he has severe GI symptoms. So what do you do with that? So up until now, because of the supportive nature of so many of the studies and slides that you've seen today, I've been treating them as we treat Crohn's disease in general. And that's with any sort of combination of immunomodulators, corticosteroids, immunosuppressants, anti-inflammatories, or biologic agents, and also diet. Diet ends up being extremely important, and that's perhaps a great topic for another time. So our experience has been that as we treat this GI enterocolitis in autism, and you see improvement in their GI symptoms, their abdominal pain, their diarrhea, their constipation, their failure to thrive, as we see improvement in those GI symptoms, that we do see improvement in those symptoms with administration of the Crohn's disease pharmaceutical agents. But together with that, the experience has also been that we see improvement in their behavior and cognition. And that the two seemed, over the years, in my experience, the two have seemed to go together. When you get a GI, the expected GI inflammatory improvement with the medications that we know help in Crohn's disease, to our surprise, we also were seeing improvements. Parents were reporting improvements in behavior and even cognition. And that was, of course, unexpected. Both of these observations have yet to be scientifically demonstrated. And the Brain Foundation has very generously funded a study to look exactly at that. And the study is designed to determine whether the GI symptoms improve with standard IBD therapy, whether cognition and behavior improves in tandem with the improvement in the underlying bowel inflammation, and also the nature of the relationship between GI symptom improvement and cognitive and behavioral. It's very interesting. Each patient is unique, and sometimes you'll see the degree of in tandem that you see the improvement will vary, and then you have to go back and wonder exactly what's happening. And that's been very instructive with so many patients over the years. And most importantly, if this correlation can in fact be statistically, mathematically, and scientifically established, meaning the association between improvement in GI symptoms and cognition and behavior, then one could say that treating underlying GI disease is an additional way to decrease the symptoms, improve the functionality of autism. And of course, that's very exciting. Briefly, the study design, we did 20 prospective first-time ASD enterocolitis patients, meaning patients who have a diagnosis of ASD and that on biopsy or endoscopy or capsule endoscopy have been shown to have enterocolitis, and treat them the way we normally would treat Crohn's patients, so no different than we had been doing for years. And the outcome measures would be, number one, improvement in the GI symptoms as a primary outcome measure, and a secondary outcome measure would be improvements in behavior and cognition. Just very quickly, I'm coming to the end. The challenge here was to create a GI questionnaire that was autism appropriate. The existing GI questionnaires that we use in academia are some of which are designed to measure objective GI symptoms like stool frequency, stool texture, etc. But many are also, many of the questions in standard questionnaires are asking the patient to tell them about specific feelings, levels of pain, intensity, location, frequency, etc. And that, of course, we wouldn't be able to know from patients who are poorly communicative. So the questionnaire was designed based upon our experience of how our patients over the years have manifested GI symptoms in a behavioral fashion. And by definition, the questionnaires have not been validated. They've been validated by our experience over the years. One thing I can say is that, and the validation work of the questions, I think will end up leading to someone else in the future. But the first step in validating them is to see that if in fact the answers, responses to questionnaires pre and post treatment, I mean, initially and then over the course of a year, if those or many of those behaviors that we think are actually manifestations of GI symptoms, if they get better in tandem with the actual GI symptoms, the stool frequency, that's the first important step to the process of validating the questionnaire. And with that, I'm going to pass the baton over to my good friend and colleague, Dr. Walker.
So Arthur, I think you need to turn your screen off, and then maybe Paul, let me start sharing.
I think it says the assessment of impact of standard care. That seems to be out there, ready to share.
Yes, but that's
there we go. There we go.
Oh, that's yours. Okay.
Okay. There we go.
You should be able to try and, yeah, there you go. Great.
Okay. Are we good to go? Okay, good. All right. Let me get this view straightened out here. Well, okay. So I've been working with Arthur now for going on almost 20 years, and I've known Paul almost as long. And I'm a non-clinician, but I've been interested in this question about the GI impact on autism and behaviors and so forth. And in the course of meetings now for the last many years, we hear a lot of anecdotal information about how if children are treated for their chronic GI symptoms, not only the GI symptoms improve, but also the ASD cognition and behaviors sometimes improve, and sometimes dramatically. But up to the point where we began this study, I believe most of this information was anecdotal. And so it was difficult to get a handle on exactly, and the question I'd ask Arthur over the years many, many times is, of all your patients, and now we have bank samples from 1,500 patients, the question I had was, who gets better and by how much. And so what we're going to do in this part of the talk is just present to you some of the compiled data from the first phase of this treatment study. So the research question is, as Arthur pointed out, does treating the ASD-associated IBD by standard of care treatment, much like you would treat someone with Crohn's disease, that is with corticosteroids and non-steroidal agents, immunomodulators, and all this is clinically warranted, does it result in improvement, number one, in GI symptoms, and number two, as an ancillary benefit, does it improve cognition and behavior in these children? Okay, so the study design, which Arthur sort of alluded to, the first step is to have histologic or endoscopic confirmation of this enterocolitis in the new patient and a GI symptomatic child with a confirmed diagnosis of autism. And then treatment, as we pointed out, with induction steroids followed by additional agents as clinically warranted. And then finally, the final step is to evaluate all along the way, both at baseline and at multiple time points throughout the course of treatment. So longitudinal data for 12 months from patient-filled questionnaires that assess both the GI symptoms and behaviors, as well as the ASD symptoms and behaviors. And so, as you can see from the chart on the right, the GI symptom questionnaires, there are two of them, and these were developed by Dr. Krigsman. And we have named these the GISS, so that's Gastrointestinal Symptom Survey. And the first questionnaire is Bowel Movement Chart Survey, and the second is Externalizing GI Behaviors Survey. So at each time point, baseline and weekly, believe it or not, for 52 weeks, the parents would fill out these two questionnaires on their child who was being treated by Dr. Krigsman. And then to look at features of ASD to determine whether there was improvement, we used three of the standardized questionnaires, the Aberrant Behavior Checklist, the ABC, the Social Responsiveness Scale, and the Vineland. And so the Aberrant Behavior Checklist was assessed at baseline and then at 3, 6, 9, and 12 months. The SRS was assessed at baseline at 6 months and then at the completion at 24 months, or sorry, 12 months. And then the Vineland was at the beginning and at 12 months. So just to recap, the gastrointestinal related questionnaires, these are the primary outcome measures. Do GI symptoms improve? And to measure this, we looked at these two questionnaires that I just described, and then three of the autism related questionnaires. And so what I'll present next is data compiled from actually going through all the answers on these questionnaires. So the GI questionnaire data, at the time that we did the data analysis, which was very recently, 11 participants, and Dr. Krigsman mentioned our goal is 20, or maybe even as many as 30. There is some dropout rate and so on. But we've had 11 participants now that we have data from baseline all the way out to 52 weeks. Now, that's not to imply every person filled out every single questionnaire, but we have a massive amount of data. So we have, for this analysis, 426 individual bowel movement chart questionnaires that were scored for this analysis. And we used the non-parametric Wilcoxon sign rank test to compare the data between time points. And this is a statistical test that measures paired data sets. So in this case, we're looking at 2 time points. It's baseline and then some subsequent time point, whether it's 3, 6, 9, or 12 months later. And then in every case, a P of 0.05, less than or equal to 0.05, was considered statistically significant improvement. And so the GISS bowel movement chart questionnaire, the first of the 2 questionnaires, consists of 17 questions. And to do the analysis, we categorize these in 5 different groups. So the first group of questions, 1 through 5, assess the stooling behaviors such as bending forward, screaming, overt straining, hyperactivity during stooling, and so on. The second set of questions, 6 through 8, we're actually characterizing the stool itself. Questions 9 through 15 are all assessed frequency between 0 and 7 days of how frequently have a bowel movement and so on. Question 16 was the Bristol stool chart, and then question 17 was another stool characteristic data point. And so you're going to see a lot of these plots in the following slides. So we'll start here. This is the analysis of questions 1 through 5 in stooling behaviors. Now, recall that for these GI questionnaires, we looked over 52 weeks. And so for some of these patients, we actually had 52 data points. For others, we had fewer than 52, but we had a large data set for each of the 11 participants. And so it would be unwieldy to present all that information here. So what we did is look at baseline, which is time point 0 on the x-axis, and then 12, 24, 36, and 52 weeks. So in other words, the questionnaire that was filled at those time points. And that will give you a very fair idea of what the trends are throughout the year. And so on the left-hand, the y-axis is the score. We set the questionnaires up so that they could be scored easily from 0 to some number. And in all instances of the slides that you'll see for these GI questionnaires, a higher number means you have a worse condition, if you will, and a lower number indicates improvement. So what you hopefully can see from this, between week 0 and 12, so after 3 months, there was significant improvement. A single asterisk at the top that signifies the comparison. A single asterisk indicates a P of 0.5. 2 asterisks will indicate even more significant, a P of 0.01. So you'll see some of those along the way. So in this case, you can see between week 0 and 12 and week 0 and 36, there was significant improvement. And overall, the trend was that the stooling behaviors improved all the way out to 52 weeks. The plots on the right are actually the individual trajectories, so you can see how they look person to person. In the second set of questions, 6 through 8, these are the stool characteristics. There was significant improvement in stool characteristics at every time point that was compared back to baseline. Questions 9 through 15, frequency 0 to 7 days of specific events. No change, no significant change from week 0 out to week 52. And I'll point out here, but I'll point out again later, that we have 52 data points for most of these children, or 40 to 52 data points. So we could get more granular with the analysis, but right now what I'm displaying for you is the overall trends. If we look at the composite score, so questions 1 through 15, now everything you just looked at, with the composite score in this case, there is only a significant difference between week 0 and 3 months out. As you can see from the plots here, there's trends. The trend is still holds that there's change and reduction in the score for most participants all the way out to 52 weeks, but it's not significant. But I would like to point out, so we have 11 patients. We're using a non-parametric test for which the indication is you really should have an N of 20 or more paired samples. And obviously in this case, we have 11. So we're a little bit underpowered in that way. And I want to show you, if you remove, there was one patient who was particularly refractory to treatment. So instead of trending downward the entire time, he trended upward. And in fact, had a difference in score of about 20 from beginning to end. And as you can see on the y-axis, a difference in 20 in the wrong direction is a huge difference. And so if you remove, and so Dr. Krigsman told me this morning, that particular patient was so impacted at a later point in the study that he had to manually unimpact this patient. So it was a severe case, and he's now being treated again. But if you remove that one patient, so now we're looking at data from 10 instead of 11, you see a significant difference pretty much all the way across the board and all the way out to 52 weeks. So that's just to say that we believe that when we have an N of 20 patients to look at, or 30 patients, these data will be more believable, but I will say they're very encouraging, even with a small N of 11 patients. The Bristol stool chart, question number 16, it wasn't included in the composite analysis because it's sort of a separate entity. And for those of you that are not familiar with the stool chart, basically, I believe the The wisdom is a type 3 to type 5 is relatively normal. Type 1 and 2 are more indicative of constipation, and 6 and 7 more indicative of diarrhea. But what you can see from the chart here is that between baseline and week 52, as a group, the patients improved significantly in a reduction from about 4.5 down to 3.5 composite score. Okay, so the second GI questionnaire measured externalizing GI behaviors. And this is one of the aspects that makes the questionnaires that were generated by Dr. Krigsman so unique, because you don't pick up these types of questions in the standard pediatric GI questionnaires that are currently in use. And so he designed these that ask questions in a way that's very specific to this particular population, because they have very specific behaviors unique to kids with autism. So again, same 11 participants. In this case, we scored 450 individual questionnaires and used the same statistics and the same significance parameters. This questionnaire, there are 3 subscales. The first subscale measures physical and behavioral signs, and that's the bulk of the questionnaire, 19 questions. The second subscale looks at extremes of behaviors, 8 questions there. And then the final subscale, it's a single question, 2 parts that ask about abdominal pain and do you have it, and how intense is it? And so if we look at the change over the course of the study for physical and behavioral signs, you can plainly see that there was a highly significant improvement from baseline to 52 weeks and at every time point in between for these 11 patients in terms of physical and behavioral signs. The extremes of behavior were significantly improved up to 36 weeks and still trending towards improvement, but you can see just looking at the plot on the left or the right, there's one time point at 52, or there's one patient data point at 52 that's way outside, it's an outlier in other words, so that's one that did not improve. That's the same patient from the other symptom surveys, but we kept that in for this. If you look at abdominal pain, significant improvement at every time point compared to baseline in the 11 patients. And again, these are comparing the individual patient data point at baseline to that same patient's data point at a later time. And we're looking at trends across the group. We're not specifically saying this patient or that patient got better at every time point or worse at every time point. It's a cumulative. As a group, there was improvement in the 11 patients. And then finally, if you look at the composite scores for the gastrointestinal symptoms, both questionnaires, there was significant improvement across the board in these 11 patients. If you look at the ASD questionnaire data, it was the same 11 participants. This time there are fewer questionnaires because, as you recall, we did the ABC questionnaires. The parents filled those out at 5 time points, so baseline, 3, 6, 9, and 12 months. So we had 50 individual ABC questionnaires. The SRS was filled out at baseline, 6 months and 52 weeks, so at the end of the study. So there were 31 of those, and then 22 individual, once at baseline and once at completion of the study, VABs. And same statistical test used and the same parameters. For the aberrant behavior checklist, the ABC scores, there was significant change, improvement from baseline to 24 weeks and also from baseline to 52 weeks. So the trend across the board was improvement and significant improvement in the ABC scores. The composite scores for the SRS, looking at 0 to 24 weeks, there was a significant reduction in the social responsiveness scale score. However, this didn't hold to 52 weeks. So from baseline to 52 weeks, statistically, they were not different. I will continue to remind you, these are 11 patients. And so I think when we have a larger data set, that some of these differences may become more clear and statistically relevant. The Vineland Adaptive Behavior Scale, interestingly, the Vineland composite scores did not change from baseline to 52 weeks, much at all. However, there are 2 components of the Vineland that measure maladaptive behavior. So in the Vineland Adaptive Behavior Scale, a higher score means you've improved. In the maladaptive subscales that are optional. Not everybody uses those. In the maladaptive behavior subscales, a lower score indicates improvement. So what we found, the maladaptive behavior internalizing from week 0 to 52, there was significant improvement in the group. And also with maladaptive behavior externalizing, there was significant improvement across 52 weeks. And this actually tracks well and maybe provides some additional support for Dr. Krigsman's externalizing behavior GI questionnaire, because the two of those I would think would be highly correlated. And so in summary, the GI surveys showed pretty much significant improvement at every time point from baseline out to 52 weeks following and during and following treatment for GI symptoms. The data summary regarding the ASD characteristics, the ABC composite scores improved across the time. The SRS did not. And the VABs did not improve as a composite, the adaptive behaviors, but in both cases of the maladaptive behavior, there was improvement at 52 weeks. And so I guess the key takeaways that we would like you to have from this are, first of all, these are preliminary results, but they demonstrate significant improvements in both GI and ASD symptoms and behaviors during and following 1 year of treatment for ASD-associated enterocolitis. As one would expect, the outcomes vary between individuals, both in the direction of change, whether it's positive or negative, and the magnitude. And if you've been listening at all over the last many years about the clinicians talking about treating children with GI symptoms and autism, this would not surprise you at all, because some children, even Dr. Krigsman's experience, continue to be refractory. These are not, by and large, easy cases. These are hard cases. That's why it takes people like him who have been at it for 20 years to have success, because you have to keep at it and you have to have a deep understanding of what you're looking at here. So the final takeaway, I believe, is we believe at the individual patient level, we have a lot of interview data here. And so a more granular look at what these symptom questionnaires are telling us, both based on the GI symptoms and the specific ASD individual domain data for these three questionnaires, in the context of the clinical picture at the times at which the questionnaires are being administered, may provide, during the course of treatment, important new details and inform the ongoing treatment strategy. Be helpful, in other words. And so finally, I can't leave without giving kudos and great thanks to Peyton Lee, my clinical studies coordinator, and Dylan Wolf, who's a first-year resident in my group. They were a terrific help in getting these data compiled. And with that, I'll end.
Thanks, Stephen. Thanks, Arthur. I think we're running a little bit behind time. So if you have an opportunity to look at the Q&A and answer those questions there are several that have been posted, that would be fantastic. And I think we can move on to the next speaker, which is Dr. Alicia Fasano. And he, in Crohn's disease many years ago, before I started moving into autism, and he had made some amazing studies in celiac disease, and particularly he was starting his work on zonulin and how that could affect barrier function. When I heard he was moving into autism research, I was really excited, because this is someone who is, like I say, a world-renowned scientist and very well established. So it helped to have some of his caliber in doing the work with autism. So that was fantastic. And on a different note, he is a fantastic singer. Not that we will get a chance to hear that, but I have had the privilege of hearing him. All right, thank you, Dr. Fasano.
Thank you, Paul. No singing today. Thank you so much. Very kind introduction. Let me start to make a quick remark. I've witnessed in this past day and a half phenomenal science. And this was only made possible thanks to the support of the Brain Foundation and the donors that generously been, donating to allow all of us to really create this syndicate of, high risk, high rewards, line of science that otherwise would not have seen the light of the prime time that we heard during this day and a half. So thank you again, Brain Foundation. Thank you, the donors, past, present, and futures that will allow us to do this kind of science.
Thank you.
Thank you, Pramila, to make us, recipient on this, projects. Today, we will talk about a theme that I heard, recurrency, this triangulation between the gut, mucosal biology and immunology, and therefore antigen trafficking, the microbiome and immune system that affects this communication, bidirectional communication between the gut to the brain. And who's going to deliver our project is a very promising colleague of mine that I believe has all the rights to, eventually make the presentation of our project, that Dr. Marcia Kinsbury. She's an assistant professor of pediatric Harvard Medical School. She's also an assistant investigator at Lurie Center for Autism at Mass General Hospital, and also one of our most brilliant principal investigator in mucosal immunology and biology research center at Massachusetts General Hospital. Her line of research has been focused on the hormone oxytocin at birth and development nervous GI system, and she's also very much interested in genetic environmental risk factors of autism. In fact, how this will affect offspring in terms of gut and brain development. And lately, thanks to this project, the Brain Foundation, she's moving toward this gut-brain axis and the interaction between the microbiome and neurodevelopment. I have to say this is complementary to a much bigger project that we have. This called GEMMA. There is a birth cohort of infants at risk of autism that will eventually fall over time, and with the multi-omic analysis and many of the stuff that you heard today. And I have sadly to report that we have recruited 100 kids, and 3 of them already have developed autism. So the incidence in this cohort is 1 in 33. This is very scary. So we hope that our science and science, the others, will try to slow down, if not stop this trend. So with that, Marcy, the floor is yours.
Thank you very much, Dr. Fasano. Can everyone see my screen?
Yes.
Okay. I also want to thank the organizers for the opportunity to speak on behalf of Dr. Fasano and our work today. I'm going to be talking about our 2-pronged approach using a humanized mouse model and human gut organoids to exploit and interrogate the zonulin pathway for personalized treatment of autism spectrum disorder. I don't have any disclosures related to this work, but I do again want to acknowledge the Brain Foundation and the O'Sullivan Foundation for their generous support of the experiments that I'll propose and present to you today. So as we all well know, ASD has core symptoms that include impaired social behavior, impaired language and social communication, restrictive repetitive behavior, as well as increased anxiety and sensory deficits and sensitivities. I also want to mention that there are comorbid diagnoses with other disorders that are seen in individuals with ASD that are relevant to some of the research I'll present today. So that individuals with ASD, approximately 30% of them are also diagnosed with social anxiety disorder and attention deficit hyperactivity disorder. And as we've all heard in this symposium over the weekend, autism is increasingly recognized and has been recognized more and more as a whole body disorder with comorbid symptoms. And the ones that I'll be focusing on today are GI symptoms, which often lead to or associated with dietary and feeding issues, as well as immune dysfunctions, and has already been nicely iterated. These GI symptoms can include constipation, diarrhea, gastric reflux, abdominal pain, and visceral hypersensitivity. And they are associated with these symptoms in ASD individuals of irritability, social withdrawal, stereopathy, hyperactivity, and language regression. And as you've also heard, the severity of the GI symptoms can, in some individuals, correlate positively with the severity of the core behavioral deficits. So that led us to, again, further address whether there's a role for gut dysfunction in the gut microbiota in ASD. And I think in listening to the talks, this is not necessarily a question anymore, but GI symptoms and altered microbiota are observed in anywhere from 25 to 70% of children with ASD. In a small open label clinical trial, microbial transfer therapy was shown to improve both the GI symptoms and the ASD core behavioral symptoms in children with ASD with severe GI dysfunction when looked at 8 weeks post-treatment, and remarkably, the benefits persisted at the 2-year follow-up in these children. In addition, targeted microbial therapy has been shown to improve specific social behavior deficits in a variety of mouse models of ASD, and shown here in this paper by Segreti et al., they looked at both genetic, environmental, and idiopathic mouse models of ASD. So, what is the mechanism by which gut microbes or dysbiosis may contribute to core behavioral symptoms of ASD? And we would like to hypothesize that a leaky intestinal barrier allows the passage of pathogenic microbes, antigens, and food particles into the bloodstream, triggering inflammatory processes, immune activation or dysfunction, and altered social behavior. So when we look, we can find studies that have reported that individuals with ASD are characterized by increased intestinal permeability. And one mechanism that we're drilling down on for the increased intestinal permeability is the presence of the haptoglobin 2 allele, or the HP2 allele. And this is an allele that's linked to zonulin production, as zonulin has been identified as the precursor protein of HP2, or the proprotein. And while human haptoglobin exists as 2 common alleles, HPA1 and HP2, generating 3 genotypes, the presence of the zonulin gene is more often encountered in chronic inflammatory diseases. And when we look at individuals with autism, this haptoglobin genotype analysis revealed a trend for overrepresentation of the HP2 allele in individuals with ASD compared to healthy controls, and an overrepresentation of the HP2 genotype and an underrepresentation of the HP11 genotype in ASD patients, specifically with GI symptoms. So zonulin is the only human protein to date that has been shown to regulate intestinal permeability by modifying epithelial tight junctions. And zonulin causes tight junction disassembly through proteinase activating receptor 2 transactivation of epidermal growth factor receptor. And this cytoskeletal rearrangement and subsequent disassembly of the tight junctions is caused by zonulin occludins 1 and occludin moving away from the tight junction, creating leaky barriers. Thus, an HP22 genotype may create the greatest vulnerability for leaky barriers due to enhanced zonulin production. And zonulin has been shown to contribute to the pathogenesis of several inflammatory diseases associated with tight junction dysfunction. Shown here in this table are chronic inflammatory diseases associated with zonulin. I highlight in here the blue that are common inflammatory diseases of the bowel. In red are common neuropsychiatric and neurodevelopmental disorders associated with some form of zonulin involvement, and autism is highlighted here. Additional evidence to support the role of zonulin in ASD comes from measurements of serum levels of zonulin, which are elevated in ASD patients and correlate positively with ASD severity as well as GI symptoms in 2 recent studies. So in the first study, there was almost a 200% increase in serum zonulin in the patient ASD group compared to the control group. And in this 2021 study, autism severity as measured by a CAR score, or the Child Autism Rating Scale, correlated positively with serum zonulin levels. Now, it's known that both gluten and dysbiotic intestinal microbiota can trigger zonulin release in the gut. increasing the transcellular trafficking of bacterial metabolites and dietary antigens. And once released, zonulin can also be transferred to the brain, where it can increase blood-brain barrier permeability and trigger neuroinflammation. And neuroinflammation and increased blood-brain barrier permeability have also been described in ASD patients. So a team member, Dr. Maria Fortino, looked at postmortem tissue from individuals with ASD and found alterations in blood-brain barrier tight junction proteins and neuroinflammatory markers. And I'll show some of that data here. So she did find that when she looked at some of the tight junction proteins, such as claudin-3, claudin-5, and claudin-12, and tricellulin, she found that compared to healthy controls, postmortem tissue from individuals with ASD showed an increase in these tight junction proteins. In addition, she found increased expression of blood-brain barrier proteins associated with neuroinflammation. And two of these proteins were MMP9. MMP9 is a matrix metalloproteinase that cleaves extracellular matrix components and is believed to be involved in the pathophysiology of neurodevelopmental disorders. And studies have shown that this protein, the secretion of this protein can cause blood-brain barrier disruption. TSBO is a translocator protein located to the outer mitochondrial membrane and has been a target for neurodevelopmental and neuropsychiatric disorders. So as I mentioned, in addition to these blood-brain barrier tight junction protein alterations and alteration neuroinflammatory markers, we know that the bacteria and microbes within the gut of ASD patients can be altered. But it is currently unknown whether the gut microbiota from ASD patients with GI symptoms exacerbates the intestinal and blood-brain barrier permeability to contribute to core behavioral symptoms. So our hypotheses for the experiments I'll describe to you today that are being supported by the Brain Foundation is that a genetic predisposition for increased gut permeability conferred by an HB22 genotype and consequently an ability to produce zonulin synergizes with dysbiotic gut microbiota from ASD patients to induce leaky gut and blood-brain barriers, producing neuroinflammation and behavioral deficits. So I'd like to introduce you to our zonulin transgenic mouse and how we're using this mouse to demonstrate zonulin-mediated increased antigen trafficking that can lead to chronic inflammation. So wild-type mice only express the HP1 allele. So to examine zonulin-dependent increased permeability, we acquired a mouse model in which the native mouse HP1 allele was substituted with a murine HP2 allele via targeted insertion to generate mice of the HP22 genotype. When we look at these mice at their small intestine and their colon, they have preserved histology, but they have increased gut permeability as assessed by a fixie-dextran assay, where mice gavage the dye, and then we look at how much of the dye is in the blood, and that's indicative of how much of the dye is transferred through the intestinal barrier. And as you can see at baseline in both females and males, the transgenic HP2 mice show increased fixie-dextran serum levels as compared to the wild-type controls. In addition, when we look at their gut microbiota through PCA analysis, we can see that ZTM mice, their gut microbiota, both the males and females, is clustering separately and differently from that of wild-type. And when we look at the relative abundance of certain species, we find that ZTM mice show gut dysbiosis with increased pro-inflammatory Rickenella and decrease in Akkermansia, which is a protective health-promoting gut microbe. We can also push the phenotype by administering dextran sodium sulfate to induce colitis in the drinking water of these mice. So when these mice are given DSS in their drinking water for 7 days, and then that treatment is stopped, a few days later, we can see 70% mortality within our HP2 zonulin transgenic mice given DSS, compared to all the other mouse conditions. And following this increased mortality, we see DSS-induced permeability in ZTM mice. This is at baseline, not under conditions of DSS treatment, but the permeability is significantly exaggerated in our Zonulin transgenic mice when they're given DSS. We can look at the histology of the small intestine, which looks relatively normal, but we can see DSS-induced colonic damage and inflammation in both our wild type and ZTM mice, but it's much more pronounced in ZTM mice. And it was interesting because in the wild type mice, one group showed persistent colitis with occasional mucosal erosion and crypt distortion, whereas another group of our wild type mice showed complete recovery after DSS treatment. But in the DSS treated mice, there was a homogeneous and severe inflammatory process with severe mucosal erosion and a loss of the regenerative crypt department. Quite remarkably, when we administer zonulin inhibitor AT1001, we can prevent the mortality and we can ameliorate colitis and intestinal permeability following this DSS treatment. So shown here is the original mortality that's induced with DSS. And when mice are administered DSS, but also the zonulin inhibitor, we can rescue that mortality. The blue line is now up here at 100%. We can also rescue the colitis through the administration of this inhibitor with the DSS treatment. And we can rescue the increases in permeability. So this again is the fixie-dextran levels in the blood under DSS conditions in our zonulin transgenic mice and in the DSS conditions with the inhibitor. I'd now like to move on and just talk to you about what we see in terms of behavior in our ZTM mice at baseline. And we've just recently published a paper in Press and Tissue Barriers showing the behavioral phenotype of these mice. So when we look at restrictive repetitive behavior as assessed with the marble burying test, the number of marbles that they bury that are placed on top of their wood chip bedding, we see a significant increase in marbles buried in female ZTM transgenic mice compared to wild type, suggesting an increase in repetitive behavior. And when we place the mice in the elevated zero maze and measure the time in the open arms versus the closed arms, we see a significant decrease in our male ZTM mice for percent time spent in the open arms, suggesting increased anxiety. We've also placed our mice into an open field arena to look at general locomotion as well as measures of ability to spend time in the center versus around the sides. And while we see an increase in the time in the center in males, when we look at the traces for males and females, we think this time in the center is just due to an increase in locomotor activity. And it seems that the traces are much more enhanced and pronounced in both males and females than that's shown by non-significant increases in distance travel. So it seems that these mice are hyperactive, and these are all under baseline conditions. Alba, within the lab, together with Maria, went on to look at the expression of blood-brain barrier tight junctions, and they found that in both male and female ZTM mice, compared to control mice, you find an alteration in the expression of tight junction proteins in males and females, occludin, VE cadherin, as well as in females, claudin 1, 3, and 5. And then in males specifically, they found an increase in two pro-inflammatory markers, IL-1 beta and CD11B. And I wanted to just mention that they focused in on claudin 5 protein expression in ZTM mice. And claudin 5 is a tight junction protein that's endothelial specific at the blood-brain barrier and is the most abundant tight junction protein. And here they found a significant increase specifically in ZTM transgenic male mice. And when Maria looked in the postmortem ASD tissue, she also found a similar increase in claudin 5 as compared to healthy controls within the cortex. So to summarize, before I move on to the experiments that we're directly working on, The ZTM mice are characterized by increased intestinal permeability and altered expression of gut and blood-brain barrier tight junctions, and increased expression of some neuroinflammatory markers and increased anxiety-like and repetitive behavior. And to summarize some of the human ASD data that others, as well as we, have discovered, individuals with ASD can be characterized by increased intestinal permeability, elevated levels of serum zonulin, altered expression of gut and blood-brain barrier tight junctions, and increased expression of some neuroinflammatory markers, and gut dysbiosis, as well as GI symptoms. So again, the hypothesis that we're testing with these experiments is that a genetic predisposition for increased gut permeability, the HP22 genotype, synergizes with aberrant or dysbiotic gut microbiota from individuals with ASD to exacerbate a leaky gut and blood-brain barrier, promoting neuroinflammation and behavioral deficits. So to address this hypothesis, these are some of the experiments that are underway with generous support from the Brain Foundation and the O'Sullivan Foundation. The first aim is to determine if stool derived from ASD patients exacerbates the gut and the brain inflammation, gut and blood-brain barrier permeability, and behavioral deficits in our zonulin-expressing transgenic mice as compared to wild-type mice. So this is a schematic of our behavioral setup. We have plans to mate female and male mice in January to generate the offspring that will serve for this experiment. We may enact an early life stress, maternal separation, to induce or to kind of push the phenotype similar to the DSS exposure. We will then perform antibiotic treatment to deplete the gut microbes in our zonulin transgenic and wild type mice, followed by fecal microbial transfer of stool collected from children with ASD with GI dysfunction and neurotypically developing controls. And so what this transfer looks like is that mice offspring of each genotype, HP11 and HP22, will receive exposure to both neurotypical pooled stool and ASD pooled stool. And as I said, we've been actively collecting stool and we're still in the process of doing that. Following the transfer, we will perform permeability assays, behavioral assays, and tissue assays of the brain, the gut, the stools, and the blood. The second aim that we're undertaking is to determine if stool derived from ASD children with GI dysfunction exacerbates small intestinal permeability, transepithelial antigen trafficking, and gut inflammatory processes in human intestinal tissue from ASD patients genetically predisposed to increased intestinal permeability, as opposed to ASD patients without this vulnerability. And so what this entails is that through a collaboration with MGH doctors for patients that are coming in for clinically indicated endoscopies, we will obtain duodenal biopsies, we will genotype the tissue, we will create intestinal organoids, we will create then intestinal monolayers where we'll perform permeability assays and inflammatory and tight junction assays. And we will do this under baseline conditions and then also following exposure to neurotypical stool as well as ASD stool. We currently have about 56 duodenal biopsies collected from both ASD and females and males, as well as a number of control males and females. And by late next week, we'll have the genotyping completed for these biopsies and be able to start creating our intestinal organoids. Perhaps the most exciting aim is to determine if the predicted exacerbation of barrier permeability and behavioral deficits that we expect to observe in ZTM mice after exposure to ASD stool can be rescued by blocking zonulin signaling using the zonulin inhibitor AT1001. So again, through the use of this inhibitor, we hope to prevent the opening and leakiness of both the gut and the blood-brain barriers. And so we will do this In the same behavioral paradigm, and the zonulin inhibitor will be added just prior to the fecal microbial transplant, and we will perform the similar assays. We will also do the same type of experiment using the inhibitor within our intestinal tissue. So here, to determine if the predicted exacerbation of barrier permeability observed in human intestinal tissue after exposure to ASD stool can be rescued by blocking zonulin signaling in our cultures using the inhibitor. And we expect the greatest exacerbation in our HP22 genotype intestinal tissue incubated or exposed to ASD pooled stool. And we expect the greatest amelioration of inflammatory and permeability by administering the inhibitor in that case. I just wanted to mention that it's exciting in that the zonulin inhibitor AT1001, also known as liraglutide acetate, is currently in use in clinical trials. It's in a phase 3 clinical trial for celiac disease in adults, and it's also in a phase 2 clinical trial for COVID-19 multisystem inflammatory syndrome in children, known as MIS-C. And MIS-C is an inflammatory disorder that stimulates a massive immune response following SARS-CoV infection. And these children have elevated levels of SARS-CoV in their stool, in their serum, as well as elevated serum zonulin, suggesting a breakdown in the mucosal barrier integrity due to increased zonulin release that may facilitate the hyperactive immune response. So it'd be really interesting to know if this inhibitor can preserve that mucosal barrier and prevent perhaps that hyperactive immune response. The proposed impact of our experiments, these are what we hope we will achieve, is that to define the role of zonulin in gut blood-brain barrier permeability in mediating the effects of the microbiota on the development and progression of core ASD symptoms. We'd like to validate zonulin and high throughput haptoglobin genotyping as biomarkers of gut-brain permeability, allowing for the stratification of the ASD population to specifically identify subgroups of ASD children that may benefit from interventions aimed at correcting gut permeability. We'd like to validate the zonulin inhibitor as an interventional tool to mechanistically link brain function and behavior to synergy between zonulin and the gut microbiota in ASD children. And finally, we'd like to provide a basis for a future clinical trial to test the effectiveness of the zonulin inhibitor in treating and ameliorating core symptoms of ASD in affected children that can be stratified for zonulin expression and/or the HP1 versus HP2 genotype. So lastly, but not least, I'd like to thank the leader of our team, Dr. Alicia Fasano, as well as past members that have been instrumental in generating the data in our ZTM mice, Dr. Craig Sturgeon, Maria Florentino, and Alba Miranda Rivera. I'd like to just shout out to our current team assembled to conduct the experiments that I proposed today. And again, to acknowledge the generous support of the Brain Foundation and the O'Sullivan Foundation for making this work possible. Thank you very much.
Thank you very much. That was excellent. That was really very interesting work. So I think, again, we've got, maybe we're running out of time. So one of the, what you could do is please email, respond to the chats again and question and answers. Pramila, what is your view on?
Yeah, thank you so much for chairing this session, Paul. So we'll take a break till 1:30 because we are running late, but sorry about that. So we'll resume at 1:30. And I wanted to mention, we keep putting up these slides with information about if anybody wants to contribute to the Brain Foundation or support this research. We have a set of greeting cards that we can send to you. So please look at that. Thank you.
Great, thank you.